Our next speaker is Gregory Farage. He's instructor in the Division of Biostatistics in the Department of Preventive Medicine here at UTHSC, and he's going to talk about comparison of linear mixed model genome scans using individual level data versus strain mean data.
Thank you, Shonak. So, oh. Okay. All right. So this is my first presentation at the CTC. So I'm glad to be here. I want to thank like the organizer and all the committee who organized like this lovely conference. So when we use like by using a strain panel, like for example the BXD family mice, we are able like to study multiple animals per strain, and by increasing the replication, the biological replication, without any additional genotyping, right? So this is one of the main advantage. But now maybe someone came to you and asked the question, should I use the individual data strain, individual data or strain mean data for my QTL map analysis using a linear mixed model? Or maybe you asked the question yourself, or maybe you didn't, but today I will try to convince you of the answer. So we have, on one hand, we have the individual data, and we have pro and con. So the main pro, it is like the individual data is a lossless data. We have all the information, and we will apply the models. All the information is here. The con, it is like it's a much high, it needs like a much higher data storage, and it can be also computationally less efficient, so slower. On the other hand, like the strain mean data, so we reduce the storage like tremendously, and also we can gain in the computation efficiency. But the con, it is like we lose some information, and by that I mean like we lose like the within variance within the strain. So potentially it could lower the statistical power, and that's what we will see. So I will not stay too long to these slides because Zifan already like have a similar one. So, but I want just to point it out that there are assumptions that we have the normality for our variance, and we assume also that the variance is the same for every strain. So we applied the model to a real data set. We applied the model here. You have the results of in the Manhattan plot of map analysis, QTL map analysis, where we try to see if we had like any association between our 20,000 marker with the trait of body weight for a group of BXD mice who had like the CHO diet data. So when we look at our population and we see the histogram of the individual size by strain, we can see that there is like very lot of disparity. Like for example, we can see here, we have few of them with 30 individuals and a lot more with less than 10 or even maybe 5 individuals. So we are going to see if this and how it impacts like our analysis. So for that, we have on one hand like the scan, I will call it individual scan, like using the individual data set. And we have like 3 different scans. I will call the regular scan, which is like using the strain mean data. And we will have the weighted strain mean, like the mean will be weighted by the number of individuals or animals per strain. And we will have the weighted with priors. So one simple explanation of the weighted with prior is that basically in addition to the mean, we use also the standard deviation within the strain. A more detailed explanation, it is like when we try like to estimate the variance in our model, we used we try to maximize we use the maximization likelihood. But we use, with this method, we use a Bayesian maximum a posteriori approach, where we use as a prior distribution, we use a scale inverse chi-square that has two parameters, the scale and the degree of freedom. And thus, those parameters are based on the standard deviation and the number of individuals per strain. So that's one of the reasons that we use this prior distribution. So here we compare like the Manhattan plot for individuals with the regular scan. And as you can see, the chromosome 19, we have like, we have like this information here. But if you look like in the other chromosome, like some is more different. And especially like, for example, you see with the individual, we used to have like here at chromosome 13 some information, but it's gone. But is it significant or not? So here we use the weighted version. So also it's kind of similar. We have a little bit more information. It's more or less similar to the individuals. And now we use the weighted version with the prior, which is, I will say, like much more similar to the individuals' results. So based just on this, and even if I repeat like many times on different traits, that will not give me an answer. We need like to have a ground truth. We need to know like which marker, okay, is real to compare. So we need like to have a simulation. So I did a simulation based on those data. And what I mean by that is like I use for the design matrix for the genotype, the real genotype matrix. I got it from Gene Networks and I didn't change it. And for the population, I use exactly the same population also from the experiment, meaning that we had the same number of strains and the same number of individuals per strain. So what changed in our simulation? We varied like the fixed effects. I mean, the fixed effects, yeah, we can vary the fixed effects. We can vary like the variance for the genetic variance and the unexplained variance. So I just want to note that we use the local approach for all the simulation. And to compare, like to have a statistical power of our simulation, we have like also the simulation under the null, so no fixed effect. So this is like what the result that we can get. So here it is like the histogram of individual per strain. That is under the results for the Y null, under the null simulation. So it's expected to have like the same distribution. So here, what is the distribution? Is the distribution of the maximum load score per simulation, meaning that here we have like 5,000 simulation. And in those 5,000 simulation, we have like each simulation, we have a maximum load score. So that's the load score for all the simulation. And here we apply like, so the different scan. So in blue, we have like with the fixed effect. And from this information, we are able like to get like a ROC curve and the AUSC. So the stronger the AUSC, so the closer to 1, the stronger the statistical power. So now, here what I have done, I kept like the unexplained variance the same, the fix is the same. I just vary from one simulation to another, just the genetic variance. So what we can see first, that is like the weighted and the weighted with prior is very close to the individuals, which has the strongest AUSC, and the regular has lower. So, and when we increase the genetic variance, we have the same tendency, even like when it is very close to 1, where the noise is, the genetic variance is random effect is very low. So now, I increase my number of simulation to 20,000, and I decrease also the number of individuals, so we have also less strain. And doing that, we have a more disparity, like differences between the different application of the scan. So we can see that here, the regular has still the lowest score, and the individual is the strongest one. And those two is very close to the individuals. So from all those 20,000 simulation, I wanted to select the one simulation who has higher heritability. Why? Because when you have the heritability closer to 1, it means that the genetic variance is stronger than the unexplained variance. And this is where we can use, have a better use of the standard deviation in our prior for the estimation of the variance. So you can see here that in those specific case, that now the weighted with prior is very close to the individual's scan. The regular scan is still much lower, and the weighted one, it's even lower. So this one, we just said like, okay, let's try like now to have like a balanced population, the same number of individual per strain. So here, I think I had like 10 or 8 mice by strain. And as expected, like all of them, they are almost the same. So there is not really information, not difference, I mean. And this is also because we assume that the variance is the same between all the strain. So in conclusion, so my recommendation, it is like the individual data scan is lossless. So it has the strongest statistical power, okay? But definitely it has like higher computation, and especially if you use the loco, you can have blotted matrix compared like to the summary data scan. So when the number of samples are uniform, like I just showed you just previously, the strain mean scan is as comparable with the statistical power of the individual data scan with more computational efficiency. So this is my recommendation. But when the number of sample is not uniform, we should use like the weighted strain mean. And if you know that the heritability is very close to 1, you should use like the prior version where you use the standard deviation along with the mean. So now you can ask the question like, what happened like if the variance is not crossed our, violates our assumptions and is not the same among the strain? Well, one option will be like to use a weighted mean variance, which is like the square root of mean over the standard deviation. But really a better solution will be like to implement a different shrinkage approach for the linear mixed model. I want to thank Sean Accents, who is actually initiate like this project and this question. I want to thank Carl Broman and Zifan for their valuable inputs, Rob Williams, Benny Mosey, Suhita Roy, and others like who participated like to get the data, and also like for the G network from where I use we used our Julia API to get all the data that was necessary for the simulation. Thank you.